Hello everyone and welcome to today's live broadcast. Hypersecretion of fibroblast-derived exosomes during chemotherapy promotes pancreatic cancer chemoresistance, presented by Reginald Hill. He is an Archibald Assistant Professor of Cancer Biology at the Department of Biological Sciences and Harper Cancer Research Institute at University of Notre Dame. I am Marjorie Torres of LabRoots and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your questions into the box that appears on the screen. We'll answer as many of your questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the button in the bottom left corner and follow the process to obtain your credits. Please join me in welcoming Dr. Reginald Hill. I will now turn the presentation over to him.
Thank you very much for the very kind introduction. Thank you to the organizers for giving me a chance to talk about my work today. As she mentioned, I will be discussing the role of fibroblast-derived exosomes in the prevention of chemotherapy resistance in pancreatic cancer. I'm the Archibald Assistant Professor of Cancer Biology at the University of Notre Dame. And so first to start, what I'd like to do is to talk a little bit about the pancreas, basically to get people oriented on what the organ actually does. So what you're seeing here on this slide is the schematic showing the normal, healthy pancreas. And what we can actually see is that the pancreas is made up of three different cell types. It's mostly acinar cells. These are the cells depicted here by the red arrow. And these are the cells that produce all of the enzymes that you use to break down your food. And the other major cell type of the pancreas is the islets. And the islets produce things that people have heard about before, such as insulin. And so the insulin-producing cells would be here, indicated by the red arrow, in the actual islets. Now, the minor population of cells in the pancreas are the ductal cells, indicated by the white arrow. And these ductal cells are the cells that control how all of these secretions and enzymes by the pancreas are actually pushed out into the body where they need to go to. And on the left, we can see here the actual orientation of where the pancreas would be. The pancreas is tucked between your stomach and duodenum. Both males and females have one, and it's a very small gland that usually is very asymptomatic, that you don't really feel, and there's no pain associated with it, even if you have a pancreatic abnormality. What we see on this slide is showing the evolution of pancreatic ductal adenocarcinoma, or PDAC. This is the most dangerous and most lethal type of pancreatic cancer that I will be talking about today. Well, what we see normally is that the pancreas has a very thin layer of ductal cells, indicated here by the red arrow, and you can see above in the schematic depicting that this very thin layer of epithelial cells is normally not very exciting. There's nothing wrong with it, and it tends to secrete the things made by the pancreas pretty efficiently. Unfortunately, what will happen over time is occasionally what will develop is a precursor lesion, or a lesion that can give rise to pancreatic cancer, called a pancreatic intraepithelial neoplasia, or a Panin lesion. As you can see here, this lesion is actually characterized by an abnormal proliferation of ductal cells, or cells that resemble ductal cells, that begin to show an abnormal phenotype in the pancreas. And finally, the last stage, the most deadly stage, is the full-blown pancreatic ductal adenocarcinoma, or PDAC. And what you can see here in PDAC, which is quite interesting, is that the actual epithelial cancer cells, depicted here by the tip of the red arrow, actually make up the minor portion of the tumor bulk. The rest of what you actually see in this slide here, this histopathology staining, is actually tumor microenvironment. So all the areas out here are actually the tumor microenvironment, the field that we actually study and what we're actually most concerned about as it relates to chemo resistance. Some interesting facts about pancreatic cancer is that now pancreatic cancer is the third leading cause of cancer death in the United States. We just this year passed breast cancer. And the interesting thing when you look at the number of cases of cancer and the various organs in the body is that although there are a lot more cases of some cancers, such as lung cancer and breast cancer, where you can see almost 200,000 people will get both lung and breast cancer every year, only about 53,000 people will get pancreatic cancer estimated this year. However, almost every single one of those people will actually die from pancreatic cancer. So we have a disease that has a very high mortality rate, so that the number of people who die from the disease is almost equal to the number that's actually going to contract the disease itself. And when we look at the actual 5-year survival rate for pancreatic cancer compared to other well-known cancers, this gives us a better indication about how aggressive and deadly this disease is. What we're looking at here is the number of patients who would be alive after 5 years of being diagnosed with these various diseases. And so what most people don't know is that pancreatic cancer has the worst 5-year survival rate of any major cancer. What we can see here is that after 5 years, outlined in the red box, only 7% of pancreatic cancer patients would actually be alive. That's even worse than lung cancer, where you can see that 17% of these patients would still be alive, or other well-known and well-studied cancers, such as breast cancer, that has a 5-year survival rate of 89%, and prostate cancer that has a 5-year survival rate of 99%. And so what we're looking at here is a disease that has a very dismal prognosis for patients who get it. And one of the reasons that this is true is because of the late stage at which the disease is actually detected. And so on the right side of the slide, you can see that when we look at how the survival rate is different based on the stage of diagnosis, most patients who have a distant metastasis, as seen here by the red arrow, have only a 2% chance of being alive 5 years after surgery. Patients who have more of a regional regional diagnosis still only have a 10% chance of being alive after they have been diagnosed with the disease for 5 years. And so this shows one of the major problems with pancreatic cancer is in that it's a disease that's only detected in its late stages. And this is because the disease is very asymptomatic. Most of the time when people go into the clinic and they complain about various symptoms, which could include back pain or they have jaundice, what they're actually describing are in fact the symptoms of the metastatic spread of the disease. So what you can see here is that only about 10 to 20% of patients are actually candidates for a surgical resection. That means they have a disease that hasn't become so aggressive and spread so far that a surgeon can't actually remove it. And even with these groups of people, their 5-year survival rate is still between median survival of 10 to 20 months. Now, unfortunately, 80% of people have what we call unresectable tumors. And these tumors are going to be already metastatic or they're already going to be locally advanced. And so this 80% of patients has the worst prognosis in that their median survival will range between 3 to 6 months or 6 to 12 months. And so part of the problem that we have is that this disease is one that's found far too late for surgical intervention, which still gives the best chance of actually curing the disease. And the really scary thing about the disease is that while it's just now becoming the third leading cause of cancer death in the U.S., it's projected to be the number 2 leading cause of cancer death by 2020, which will be second only to lung cancer. And the reason that the number of deaths attributed to pancreatic cancer are projected to go up are, again, because of the late stages of which we detect the disease and the actual incidence of the disease increasing due to a variety of factors that you can see here on the top of the slide, including smoking, obesity, poor diet, family histories, and the increase in type 2 diabetes in the country. So all of this really gives us the reason to want to really focus on understanding how to come up with better ways to therapeutically treat this disease. When we look at the most commonly done treatment for pancreatic cancer, it is chemotherapy. And so the very remarkable thing about this disease is that it's very resistant to single agent and combination chemotherapy. Gemcitabine, also known as Gemzar, is a nucleoside analog that is the most commonly given chemotherapy for pancreatic cancer. As you can see here on this slide, when gemcitabine is given to patients who have metastatic pancreatic cancer, the median survival will only be about 5.91 months. And so we know now that most chemotherapy is not as effective alone as it could be when given with other agents. However, we don't see an added benefit with gemcitabine and a variety of other chemotherapies. Even when we look at using gemcitabine in combination with something like erlotinib, we still only see a median survival of 6.24 months. Now, FOLFIRINOX is a combination of multiple types of chemotherapies, which has the largest increase in median survival, where patients will have a median survival of 11.1 months, as depicted here. However, this combination is very harsh on the patients, and therefore only people who have an unbelievably healthy profile are even candidates for this chemotherapy. And it still shows a lot of chemoresistance, and then when these patients fail FOLFIRINOX, they will go back on gemcitabine-based therapy. Nab-paclitaxel and gemcitabine was really thought to be a huge step forward for pancreatic cancer research in patients. It was something that was hoped to double at least the survival of patients. However, we see here that there's only an 8.5-month median survival for patients who are treated with both nab-paclitaxel and gemcitabine. And so still we're at a point where we have a remarkably chemoresistant disease that doesn't respond to single agent or even combination therapy. And so what we've been trying to figure out is why. So in order to really address why this disease is so chemoresistant, we first have to start at the beginning. What do we currently know about pancreatic ductal adenocarcinoma? We know that there are a variety of different genetic alterations that are found in pancreatic cancer, as depicted on this table here. KRAS mutations are by large the most prevalent mutation found in pancreatic cancer. Here in this chart, it's showing the mutation frequency at about 58%, but overall most studies would suggest that the number of KRAS mutations are anywhere between 90 and 95% in pancreatic cancer. It has one of the tightest correlations between one single genetic event and the onset of an actual cancer. And so this was really a very interesting thing to look at in terms of understanding the role that the KRAS mutation may play in the development and the aggressive nature of pancreatic cancer. The KRAS mutations that are found in the majority of cases of PDAC are actually very important because a mutation in KRAS will lock a very important signaling pathway into its on conformation. So that what you see usually happen is that RAS, which is usually able to fluctuate between a GTP and GDP form, will be stuck in a on-activated position, where a mutation in residues 12, 13, or 61 will make RAS constitutively activated so that it's always on. This leads to increased expression of genes linked to cell proliferation, migration, and proliferation. And so this was one of the first things we knew about pancreatic cancer, is that this gene, which is very important for cells proliferating and migrating, was found to be highly expressed in pancreatic cancer. And so if we believe that this gene is actually very important for pancreatic cancer development and aggressiveness, how do we actually test this? What we knew at this point was that simply this gene was actually correlated with pancreatic cancer, but how do we actually prove this gene or even other alterations are actually important in causing pancreatic cancer? And what we traditionally done as researchers is to use mouse models. I've made a variety of mouse models in my career for both prostate cancer and pancreatic cancer. I've made so many that I actually use the Twitter handle of Mouse Modeler. So if you want to follow me on Twitter and learn more about mouse models or see various tweets about the research we're doing or other research in the field, that you can do that as well. And so the very interesting thing is that these mouse models allow us to actually directly test the function of a gene. And these mouse models are great because if we can produce a mouse model that fully recapitulates the spectrum of phenotypes that we see in the human disease, we can use these mouse models to evaluate strategies to detect these cancers early, or more importantly, come up with ways to treat these cancers effectively and then to translate those findings onto patients. And so what people did was to actually create a mouse model using a mutated version of KRAS. So what we do here is to actually express KRAS, a mutant allele, directly in the pancreas of transgenic animals. And the surprising thing was that when we introduced this mutant allele of KRAS into mice, researchers found that there was only the progression to pancreatic intraepithelial neoplasias, or PANN. Remember what I said earlier, these are the precursor lesions that lead up to full-blown cancer, but this is not actually pancreatic ductal adenocarcinoma itself. And so we found that these mice will very rarely, and only after a very long time, progress to full-blown PDAC. This suggested that while these KRAS mutations were very important for causing precursor lesions, they were actually not sufficient to give the full spectrum of tumors that we saw in patients. So this really raised the questions of what's missing from this process. What other events do we have to layer on top of this event in order to get the full spectrum of tumor formation that we see in human patients? And this is where the ability to layer one genetic alteration on top of another has really benefited mouse modelers for a really long time. And so we know that there are a variety of other tumor suppressor genes that are important in pancreatic cancer development. So even if they by themselves don't cause pancreatic cancer, what researchers were able to show is that these mutations or alterations, when added to an existing KRAS mutation, would give rise to full-blown PDAC. And we saw this in cases where people would look at things like INK4A loss, P53 mutation, TGF-beta receptor knockout, SMAD4 loss, and even cases of looking at things related to snail activation. All of these events could push a precursor lesion that's caused by KRAS to a more aggressive inflammatory state into full-blown pancreatic ductal adenocarcinoma. And so knowing this, what I decided to do was to take an opportunity to look at what in pancreatic cancer that hadn't been looked at before might play a role in progressing tumor development in the context of a KRAS mutation. So that's where I looked at the status of PTEN in pancreatic cancer. PTEN is one of the major tumor suppressor genes in the body that's responsible for suppressing the activity of things such as phosphorylated AKT. When activation of AKT is seen, we know that this leads to increased cell cycle progression and suppressing cell death. This is a major tumor suppressor gene that's mutated or altered in about 30 to 50% of all cancers. So what I wanted to really explore was whether or not PTEN played a role in pancreatic cancer progression. So in order to actually test this, what I did was to combine an animal model that gave us the ability to activate a mutant allele of KRAS and knock out PTEN in the same cells in transgenic animals. So what you're seeing here is a schematic that depicts that strategy. And so what we did was to have a floxed allele of PTEN, as you can see here by the red arrow. This critical allele of PTEN will be placed between two LOXP sites. So that meant whenever it came in contact with Cre recombinase activity, it would be deleted and removed, resulting in a version of the PTEN allele that doesn't have the full functionality of the normal PTEN protein. And we also added this to the same KRAS model that people had already used, so that what we were able to do is to have a mutant allele of KRAS that was floxed or flanked by two LOXP sites, or stop codon, sorry, is actually flanked by two LOXP sites. And then this gives us the ability to knock out PTEN and overexpress a mutant allele of KRAS in the same cell. So what this gave us the ability to ask the question, if we knock out P10, which we know didn't cause pancreatic cancer, and we combine this with a mutation in KRAS, which led to precursor lesions, what would be the actual phenotype? Would this actually give rise to a very aggressive tumor that would mimic what we saw in the human disease? And the answer we got was very surprising and very quickly that yes, it did. So what we see is that this is a mouse here on the right that succumbed to a pancreatic cancer tumor. It's only about 14 days old, and it has a very slouched posture, and the abdomen is kind of bulging here. You can see on the left side of the screen that the pancreata of wild-type mice had the normal architecture where you can see all the normal acinar cells, islets, and ductal cells. However, the mouse with KRAS activating mutation and full loss of P10 function had a completely dysregulated and abnormal pancreas, where most of the acinar cells are gone, and we see the creation of ductal structures and general destruction of the normal architecture of the pancreas. Unfortunately, these mice actually died before we could wean them. So mice with pancreatic cancer that develop so quickly are unfortunately not very useful for the types of studies we wanted to do. These mice died so fast we couldn't really study how to prevent the tumors from developing. And so what we needed to do was to develop a mouse model that developed tumors using the same genetic alterations, but actually it would develop slower over time. And so in order to do this, we simply looked at the heterozygous state of P10 as opposed to the complete loss of P10 in this genetic system. So what we're looking at here is a graph showing the percentage of mice with PDAC compared to mice with KRAS activation or mice with KRAS activation and P10 heterozygosity. And what we can actually see is that at 3 months of age, there is about 60% of the mice have PDAC if they have KRAS activation and P10 heterozygosity, compared to no mice having PDAC if they're KRAS only. And by the time the mice are 6 months of age, then we see this number only gets higher in the mice that have KRAS activation and P10 heterozygosity. And finally, at this point, we see a few mice that are just KRAS only starting to develop pancreatic ductal adenocarcinomas. This was quite interesting as we looked at why these tumors were forming in these mice. Again, we know that P10 does a big job in suppressing the activity of phosphorylated AKT. So when we looked at the precursor lesions found in both mice that had KRAS activating mutation and KRAS activating mutation combined with P10 heterozygosity, what we saw is that there was loss of P10 based on the activity of phosphorylated AKT in PDAC samples. So we can see here in the red arrow as immunohistochemistry, you can see the brown staining showing that phosphorylated AKT is up. And the reason that we believed it would be higher is because of loss of P10 expression. So in order to actually confirm this, we looked at the actual tumors that developed and performed laser capture microdissection. And what this is that you can actually take a slide where you have a tumor sample, and using the laser on a microscope, you can cut out a piece of this tumor and essentially do genotyping on it to look for the presence of wild type, alleles, LOX alleles, or the absence of those alleles. What we actually saw is that in these mice that have full-blown pancreatic ductal adenocarcinoma, and virtually all of these mice showed a loss of the wild type allele of P10. You can see that here depicted in the red arrow by the red box. The wild type allele was lost in 4 of the 5 samples that we looked at. Same thing for the precursor lesions. They generally had the wild type allele presence, but it was gone by the time that these tumors developed. And this really suggested that loss of P10 heterozygosity was important for this progression in tumor genesis. So this really helped us put together a nice story showing that P10 plays a dose-dependent role in tumor initiation and progression in pancreatic cancer. So that if we have KRAS combined with P10 haploinsufficiency, this will eventually give rise to P10 loss of function, which will lead to pancreatic ductal adenocarcinoma. So when we look at our survival curve for these different animal cohorts, what we see is that if mice have KRAS mutation and have normal PTEN function, kind of indicated by this blue box here. These animals are fine. They live into adulthood. They really don't develop tumors that are lethal at all. However, if we have KRAS mice added with the heterozygosity of PTEN, these mice start to die at about 3 months of age, and they're all dead by a year. Now, as I first showed you, if we remove both copies of PTEN, this dramatically shifts the survival curve so that these animals depicted by the red line here will actually die of their disease before we can even wean them. These animals die before 20 days of age. So what we've shown is that PTEN is a very important gatekeeper in preventing the development of very lethal tumor development in the pancreas. And so this was quite interesting as it related to how PTEN may play a role in the progression of pancreatic cancer in a mouse. What we really wanted to do was to actually explore how this could play a role in human cancer. And so in collaboration with surgeons at UCLA, what we performed was a comprehensive bioinformatic analysis of human patients to figure out which pathways and which level of regulation was important for preventing patients from developing lethal tumors. So what we examined were resectable tumors. So these are the tumors of patients who came into the clinic early enough to have their tumor samples actually collected. And then what we did was to do an array profiling looking at microRNAs, RNA, and SNP analysis to determine which pathway alteration separated these patients into good or poor prognosis. So to say patients who did well and lived longer, what was their genetic profile like compared to patients who succumbed quicker to their actual disease. And what we saw is that the PTEN pathway, and in this case through microRNA alterations were responsible for separating these patients into good and poor prognosis. So this really combined nicely with the data that we had from the mouse model, because what we could see is that the mouse model that we generated looking at KRAS activation and PTEN heterozygosity actually did a great job of recapitulating the pathology of the human disease so that the mouse and both the human samples look the same. But more importantly, on a genetic level, we had data showing that PTEN suppression of tumor development was the thing that led to people either having a good or a poor prognosis. So if you were a mouse and you had PTEN fully functional, you've lived really far, long life, as opposed to mice that did not have PTEN function. And this was the same in the humans. Humans that had PTEN functional had a better prognosis compared to humans whose tumors had a loss of PTEN function. So this really, again, cemented the role of PTEN in this process of suppressing tumor development. That was a good start for our model, but what we needed to do then was to address a major hallmark of pancreatic cancer, and that is inflammation. Inflammation plays a critical role in cancer progression in a variety of different sites. We know that lots of sites of cancer will arise from areas of chronic irritation, infection, and inflammation. And by the age of 70, approximately 40% of patients with hereditary pancreatitis will have been diagnosed with pancreatic cancer. So as we see depicted in this schematic here, we know that the risk of cancer is actually going to increase because of inflammation. So what we wanted to look at is the role of cyclooxygenase 2, or COX-2. This is the key mediator of inflammation in the body. Inflammation goes through a couple of different pathways. There's a couple of different forms of cyclooxygenase. COX-1, which is a constitutively active form of this enzyme, and COX-2, which is an inducible form. And so things such as nonsteroidal anti-inflammatory drugs, something as simple as aspirin, this will actually target COX-2, this inducible form. What we had seen in a lot of different places is that COX-2 expression, just like KRAS mutations, were very prevalent in pancreatic cancer. So what we wanted to show is whether COX-2 overexpression on its own could actually cause pancreatic ductal adenocarcinoma. I'm not going to show you that data now out of the interest of time, but we don't actually see that tumors occur when you just have COX-2 overexpression. But that raised the question of, do you have the ability to show that this inflammation driven by COX-2 will actually lead to increased tumorigenesis in an existing mouse model such as the one that I described earlier? And in fact, it does. So what we're looking at here is a chart showing the comparison of tumor phenotype in mice that have just the KRAS activating mutation in PTEN heterozygosity compared to the mice that have KRAS activating mutation, PTEN heterozygosity, and inflammation is indicated by COX-2 overexpression seen here. What you can see here on the right of the graph is that the mice with COX-2 overexpression have even more and earlier tumor development than our previous mouse model. So again, the earlier mouse model got tumors pretty quickly at around 3 months of age. What we're looking at here is a 1 to 2 month window of time, and we see that there are already full-blown pancreatic ductal adenocarcinomas that are formed in these mice that have COX-2 overexpression. Now, the really exciting thing is when we look at the actual pathology of these tumor sections under the microscope, we see something very remarkable. When we look at the mice at this stage, we see that the mice with KRAS activation and PTEN heterozygosity have precursor lesions and a little bit of ductal structure forming. You compare this to what we see in the mice with COX-2 overexpression added. They have full-blown PDAC. You can see the epithelial cells marked by the red arrow there. But more importantly, they have this massive desmoplasia. And what this is the tumor microenvironment that's surrounding the cancer cells. As I mentioned earlier, the remarkable thing about pancreatic cancer is that this tumor microenvironment makes up about 90% of the tumor bulk. A better way of seeing that is perhaps depicted here when we look at Mason's trichrome staining. And what this actually detects is collagen deposition by fibroblasts. And what we can see in that these mice that have COX-2 overexpression have very robust collagen deposition that starts even at the earliest stages in the pannin or precursor stage, and it's very robust when we have the full-blown pancreatic ductal adenocarcinoma, a lot more so than what we see when these mice just have KRAS activation and PTEN heterozygosity. And so this desmoplasia is very important in that this fully recapitulates what we see in the human disease. When I showed these slides to pathologists, they were like, that's it. These are the ones that actually resemble the pancreatic cancer that we see in human patients. And so we were able to show that our mouse with COX-2-driven tumor growth really mimics that desmoplasia that we see in the human condition. So the interesting thing again is when we compare the pannin to the PDAC, you can see on the yellow arrows here, that yellow arrow is pointing to the stroma or the tumor microenvironment fibroblast layer in the normal cells and in the actual tumors. Compare that again to what we see in the actual human sample, so that we have a lot more desmoplasia in the humans than we see in most cancer. But more importantly, our mouse model actually mimics this very nicely, which says that we can look at the role that inflammation plays, and more importantly, the role that fibroblasts play in making these tumors worse or possibly chemo-resistant. A lot of times when people talk about how, there's a root to a problem in cancer, it might be something related to cancer stem cells or resistant populations of epithelial cells. I like to think about what the tumor microenvironment, all these fibroblasts represents. And again, here, this is trichrome staining, to where we see all this deposition of collagen by fibroblasts. This creates a soil that these cancer cells actually grow in. And so I think of this as not getting to the root of the problem, but the soil of the problem. That's where we have the bigger issue. The tumor microenvironment itself is what we have to focus on in order to figure out how to make significant progress in treating this disease. One of the things we looked at when we looked at patient tumors was what happens to them when they're actually treated with either saline or chemotherapy like gemcitabine. What we're looking at here is the proliferation index by IHC. So this marker Ki67 will stain the nuclei of cells that are proliferating positively. And what you can see here is that the epithelial cells are present and they're proliferating. There are lots of little brown dots you can see in a variety of these cells. And so the interesting thing to me was that when we treated these patients, these tumors, with gemcitabine, we saw that the number of epithelial cells actually went down. There's a lower percentage of epithelial cells overall here, and you'll see that these epithelial cells actually have a lower proliferation index. They're not showing as many brown nuclei that are staining positive for Ki67. However, the thing that didn't seem to change was the tumor microenvironment. So these fibroblasts were here just as prominently or even more so than they were before the tumors were actually treated. So to me, that raised a very interesting question. Question being, why are these stromal cells, these fibroblasts, still here? if we are successfully removing the epithelial cells and not the stromal cells, might this suggest that the stromal cells themselves might be responding to chemotherapy differently, and they might actually be resistant to the drugs themselves? And so what we wanted to do was to examine the role of fibroblasts compared to epithelial cells. The fibroblasts depicted here by these little triangles, or these little diamonds, people had examined the role of normal fibroblasts as they interact with normal cancer cells or normal or tumor cells, or the role of cancer-associated fibroblasts with normal epithelial cells and tumor cells. And so what we specifically wanted to address was how chemotherapy, when it's hitting these fibroblasts that are associated with tumors, how is it actually affecting the tumor cell? And so this is really what our study really focused on. And so what we wanted to know are, is it possible that chemotherapy resistance can actually come through factors secreted by cancer-associated fibroblasts, and this could actually have an effect on the role or the ability of epithelial cells to actually resist the chemotherapy they're exposed to. And so what we actually did was to take media from fibroblasts that we had already previously found to be resistant to chemotherapy. And so when we found that these fibroblasts were resistant to chemotherapy, what we wanted to do is to see if they could actually help support the role or the growth of epithelial cells that were, sensitive to chemotherapy. And so we grew these fibroblasts, which we got from a collaborator at Indiana University in Indianapolis. We grew them, and we took the media from these fibroblasts, and we isolated it and put it onto L3.6 pancreatic cancer cells. And these pancreatic cancer cells are actually sensitive to chemotherapy like gemcitabine. So when they're treated with gemcitabine, they'll actually die. So what we saw was that when we simply added the media from the cancer-associated fibroblasts to these L3 cells, you can see here the purple bars, we had a greater number of cells proliferating. So the control that we used here was just the L3.6 cells added, their media added to themselves. So we can see is that the number of cells that were alive was higher when we used trypan blue counting or even an MTT proliferation assay. Now, the most important thing happened when we actually took this media and added it to L3.6 cells and then treated them with chemotherapy. So what we saw here is that the media from the calves enhanced the ability of these L3.6 cells to survive exposure to chemotherapy. So this was what was really exciting, because this showed that there is something that's secreted by these fibroblasts that can actually keep cancer cells alive and keep them from dying when exposed to chemotherapy. And so what we wanted to do was determine what that factor might be, or more importantly, how it might actually be released from these cancer-associated fibroblasts. And this is where we came upon exosomes. So exosomes are very exciting molecules or delivery vectors that people are looking at right now. And exosomes are very small lipid vesicles between 30 and 100 nanometers in size, and they're secreted from cells, and they contain a lot of different things, including microRNAs, RNAs, DNA, and protein. They can travel long distances in serum. An interesting thing that we'd seen is that cancer cells secreted more exosomes than normal cells. So what we really wanted to figure out is how these exosomes might be playing a role in the ability of calves to transfer factors that might promote chemoresistance in cells that uptake them. So what we did was to label a cancer-associated fibroblast cell line with GFP. So what we use is the CD63 GFP labeled cell line that we created. And CD63 is a marker of exosomes. So what we saw is that when we looked at the normal level of GFP expression in fibroblasts, we saw a very mild low level of GFP staining. However, when we increased the amount of gemcitabine, we added a lot of gemcitabine to these calves, the amount of fluorescent staining that we saw was greatly increased, so that the total fluorescence seemed to go up dramatically in these cells. We were able to actually quantify that looking at corrected total fluorescence, so that the number of GFP positive signal that we got went up a lot in the cells that were treated with gemcitabine. In order to actually show that in a little bit more quantitative manner, we use a nanosite to actually measure the exosomes that were released by the cells. And this is where we came across a very interesting finding. What we see is that the cancer cells, whether they're the pancreatic cells that are epithelial in nature, will have exosome secretion that will increase when they're treated with gemcitabine, here depicted as GT, which means gem treated compared to NT non-treated. However, the wild-type fibroblasts or the cancer-associated fibroblasts both had a lot more exosome secretion or exosome hypersecretion when they were treated with gemcitabine. So this was quite interesting. So what this showed was that fibroblasts, more so than cancer cells, had a very accelerated rate of exosome release following exposure to chemotherapy. So what we wanted to look at then was what happens to cells that take up these exosomes that have been exposed to chemotherapy and have been released by these fibroblasts. And so what we looked at was the proliferation in these cell lines that had received these gem therapy treated calf-derived exosomes. And we saw in these drug-resistant cell lines, the PANC1 and the ASP cell line, that both of these cell lines had an increase in cell number following exposure to exosomes from these fibroblasts that had been exposed to gemcitabine. And more importantly, this increased the ability of these cells to survive chemotherapy. The thing that was most impressive was that we got the most dramatic effect in these cells that had the gemcitabine treatment and were given calves that came from, the calves that had the fibroblasts, and these cells were the ASPC1 cells. So this is our metastatic pancreatic cancer cell line. So what we see here is that the calves give this metastatic cell line the most protection, and this cell is drug resistant as well. And so what we see in patients when they come into the clinic is that this is the type of tumor that they will have. It will be metastatic. It will already be resistant to gemcitabine. And so compared to the ability of these cells' own exosomes to confer resistance to gemcitabine, the calf-derived exosomes had an even greater impact on allowing these cells to actually survive, which is probably a very important clinical finding. And so having shown that these exosomes seem to play a very important role in giving these cells the ability to survive chemotherapy exposure, what we wanted to do was to figure out how to actually block this exosome release or secretion. So we identified a inhibitor of sphingomycinases, GW4869, and this inhibits exosome release from cells. So what we're able to show is that using a transwell system where we cultured the fibroblasts in the upper level of the dish and the epithelial cells in the bottom portion of the dish, we were able to show that we could inhibit the release of exosomes from these fibroblasts by using GW4869. What you're looking at on the right is the quantitation of this effect. And so normally when we treat these fibroblasts with gemcitabine, we get a nice induction of exosome releases, comparing the red bar to the black bar. However, when we treat with GW4869, even in the presence of gemcitabine, we don't see that increase in exosome secretion anymore. So we're blocking about 80 to 90% of the exosome secretion with the treatment of these cells with GW4869. And so what we wanted to do then was to figure out if blocking the exosome release from these calves would actually alter that ability that I just talked about earlier, the ability of these calf-derived exosomes to increase chemoresistance in the epithelial cells. And that's actually what we saw. So compare again here the exosomes from the ASPC1 cells. When we give these cells exosomes from calves, they have higher chemoresistance, comparing the white bar to the red bar. However, when we treat these calves with GW4869 and block the exosome secretion, then we're actually decreasing their ability to enable this chemoresistance. So we're doing a partial rescue here of this effect, which is very exciting. This shows that by simply blocking the exosome release, we're able to take away this protective effect that the calves are providing for the epithelial cells. And this really raised the question of, could we actually study this in vivo? How would this consequence actually play out if we were able to block the ability of exosomes from fibroblasts to support chemoresistance and tumor cell growth in an actual mouse model? And so previously we had looked at this in our in vitro system using transwell assay system, but now what we wanted to do was combine these fibroblasts and epithelial cells together in the natural situation where they would normally exist, where they're actually co-cultured and grown together. So what we did was to take mice and inject them with a mixture of epithelial cells and fibroblasts and plant this into the mice subcutaneously and let this grow for 2 weeks. And then, as you can see depicted on the schematic, we would inject with either regular chemotherapy or chemotherapy and GW4869 to block exosome release, and then measure tumor survival, or tumor growth in these animals. What we actually saw is that when you look at the red line, these are the untreated mice compared to the gray, the treated mice with just gemcitabine, whereas gemcitabine, you compare red to gray, doesn't actually show any effect on the tumor growth of these mice after 10 days. When we use gemcitabine in combination with GW4869, now we have significant decrease in tumor size compared to the untreated control. So whereas gemcitabine alone doesn't actually significantly alter tumor size, once we add the ability to block exosome release in this in vivo experiment, we have successfully blocked tumor growth to a significant degree. And so at this point, we had determined that using gemcitabine treatment would cause the hypersecretion of exosomes from calves. This would lead to increased proliferation in drug-resistant and epithelial cells, and then by using GW4869, blocking exosome hypersecretion, we could inhibit this process. But what we still didn't know at this point was what was the actual mechanism that's responsible for this. And so in order to really try to figure that out, what we did was to perform a study where we characterized the microRNAs that were found in the exosomes of calves when they've been treated either with gemcitabine, without gemcitabine, or when they were not cancer-associated fibroblasts at all, as you can see here depicted in the schematic. And what we identified were microRNAs that were differentially regulated in these different situations. And this was a study that was funded by the Indiana CTSI. And so what we did was to come up with a list of microRNAs that were being released by these calves when they were treated with chemotherapy, and then to look at the targets of these microRNAs and to figure out which ones might most closely associate with things like poor prognosis in pancreatic cancer. So we developed a short list of 5 microRNAs, as you can see here on this table. They were all significantly increased by fold change anywhere of 2.5 to 4.6-fold increase in the calves that have been treated with gemcitabine. So these were actually the microRNAs that were enriched in the calves that have been treated with chemotherapy. And the interesting thing that we found, and why we like this short list, is because all these actually were mirrors that actually targeted P10. So what you're seeing here is their score showing how closely they or tightly they regulate P10 function. So we found it interesting, again, that from our earlier studies, we'd shown that P10 was a very important regulator of tumor growth or aggressiveness in the pancreas, and that all these mirrors, these microRNAs, targeted P10. And so what this really suggested, a hypothesis, was that these calves were really aiding the epithelial cells by suppressing P10 function, giving them the ability to activate a variety of different cell survival signals. So if that's true, it should mean that we should be able to look at these tumors that we treated in these mice and see a P10 change associated with this exosome release. And that's actually what we saw. So when we look at the mice that had been treated with gemcitabine, they grew very quickly and they had very low P10 expression. However, when we looked at the mice that had been treated with gemcitabine and GW4869, what we see is that not only did this treatment actually restore P10 function, but it also, again, was the one that actually shrank the tumors or caused the tumors to grow to the least amount. So again, this really kind of explained our finding that the exosomes are probably targeting P10 function, and that's how they're actually contributing to this result. So we looked at whether or not we could simply deplete the exosomes to get the same effect. Again, kind of more proof of principle of our hypothesis. And so what we're seeing here on the top left is that these exosomes from calves, when added to ASPC1 cells, they suppress P10 expression. However, if we deplete the calf's media for exosomes, we can basically just spin out the exosomes with ultracentrifugation and then add that to the ASPC1 cells, then the amount of PTEN expression goes back up. Same thing with the L3.6 cells. Again, this is the drug-sensitive cell line where we see the exact same effect, where when we look at on the right side, the calves that are, and we take their exosomes and add it to the L3.6 cells, we see decreased PTEN expression. We deplete the exosomes from the media, and the PTEN expression goes back up. On the bottom portion of the screen, what you see are the associated Western blots that we did for that as well. So again, showing that calves with ASPC1 cells show a decrease in PTEN expression. When we deplete the exosomes from the media of calves, we see that go back up. And on the right, what we're showing the L3.6 cells is that phosphorylated AKT expression correlates with that in the way that we would expect, so that when we incubate these L3.6 cells with calf-derived exosomes, we see an increase in AKT expression, and this decreases when we do the exosome depletion from the media itself. We decided to quickly kind of explore the ability of the top hit that we had, miR92A, to carry out this process. So we developed a cell line where we could overexpress miR92A. So we used a miR92A mimic that we can get robust overexpression of miR92A, as depicted by the graph on the left side of the screen. On the right, you can see that when we overexpress this miR92A mimic, we knock down PTEN expression pretty nicely. And so what we saw is that when we look at the expression of miR92A in the environments that we first identified this effect in, when we have calves that are incubated with ASPC1 cells, these ASPC1 cells show an increase in miR92A expression, as depicted by the red arrow. And this is true in the ASPC1 cells and also in our drug-sensitive L3.6 cells. And when we look at the expression of miR92A in the GW-treated calves, it decreases as well. And so we're showing here again that this miR92A seems to be probably what's one of the key microRNAs responsible for suppressing PTEN function in this system. And so what we have been able to show so far with our proposed mechanism is that gemcitabine will cause the hypersecretion of exosomes, and that these exosomes will have a high amount of microRNAs that target PTEN. And this decrease in PTEN will lead to an increase in proliferation and chemoresistance. And so our kind of major findings are that these gemcitabine-treated calves are very important in helping to transfer the ability of chemoresistant to recipient epithelial cells, and that this role is really mediated very large part by microRNAs, and that by inhibiting the exosome signaling, as we've been able to do with GW4869 or through exosome depletion, we've been able to suppress tumor growth and chemoresistance in our in vitro and in vivo experiments. So when we look at how all of this really ties together, my lab has looked at a variety of different ways to explain how the tumor microenvironment is responsible for driving progression of pancreatic ductal adenocarcinoma. One of the things I did not have time to talk about today was the role of the endoplasmic reticulum stress, or ER stress, and how that affects chemoresistance, also something that's driven by the microenvironment. We've identified a compound that can actually suppress ER stress, and this is something that we've helped to get orphan drug designation for, and we're moving forward with that in clinical trials. We also know that the tumor microenvironment plays an important role in the ability of cancer cells to metastasize and to spread to other organs. And we've identified a molecule, MTSS1, that stabilizes the ability of these tumor cells to leave their location, and hopefully that will give us the ability to prevent the amount of metastatic cells that are found in patients. What I talked to you today most about was the exosome hypersecretion, how we know that the tumor microenvironment, these fibroblasts specifically, produce a high amount of exosomes, that these are released to give rise to a variety of different phenotypes, including increased proliferation and increased chemoresistance. And that's the paper by Kate Richards, my grad student, that came out earlier this year. And one thing I don't have a lot of time to talk about, but I wanted to mention, is that we know that these exosomes can be very important for a variety of different things. These exosomes, again, are very small. They're found in all solutions in the body, and they travel a long distance. And so these exosomes contain microRNAs, which are a great biomarker for cancer. And so one of the projects we've done in collaboration with engineers at Notre Dame is to look at how this might be very useful for early detection. So that's a study that we've already published with an engineering group on campus. I just want to take a couple of minutes to wrap up and show a few slides about our early detection of pancreatic cancer. Again, since this is a major problem that we face with why this disease is so aggressive. So what we collaborated with this group on is to figure out a way to use the information inside exosomes, these microRNAs, for early detection of pancreatic cancer. And we do this by developing a microfluidic chip that will lyse exosomes and release their contents. And these microRNAs will then be bound to a membrane sensor, and this gives us the ability to actually use these microRNAs as biomarkers so that we could detect pancreatic cancer earlier than we've ever been able to before. One of the biggest problems of why pancreatic cancer is so aggressive is that there are currently no standard screening protocols for it. There's nothing like, mammograms or other things that are done for, prostate cancer. And so part of the reason we really want to push this type of project is because we need to figure out a way to detect the disease as early as possible. And again, that's how the mouse model can actually help. We know when mouse models get precursor lesions, we know when they get tumors, so we can use our animal models to show the usefulness of using the exosomal microRNAs for early detection of pancreatic cancer. We can treat these mice with chemicals, different chemotherapeutic agents, and to look for the decrease in expression of our biomarkers in these exosomes, again, to give us not only diagnostic information, but prognostic information as well to help to treat patients better and more effectively than we've been able to do. And so with that, I'd like to end and thank the grad students in the lab and undergrads who do the work, my collaborators at Notre Dame and other institutes, and our funding sources. I'll be happy to take any questions.
Thank you, Dr. Hill, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window. Type your questions into the box that appears on your screen and click on the send button. We'll answer as many of your questions as we have time for. Let's get started. Our first question is the exosome hypersecretion seen only with gemcitabine treatment?
Yeah, the question is this only gemcitabine based? That's a great question. We first looked at gemcitabine because of the role it plays in being the predominant chemotherapy given in pancreatic cancer, but we've started to look at other compounds as well, including nab-paclitaxel, which again is the combination therapy that's most currently now given in pancreatic cancer. And we found that the exosome hypersecretion is in fact caused by almost every agent that we can find, including nab-paclitaxel. And so what it seems to be is that it's not a gemcitabine specific response, it's a response to chemotherapy or in general any type of insult or stress to the cell that causes it to hypersecrete exosomes. And so how similar those exosomes, say from nab-paclitaxel compared to gemcitabine may be, is something that we're currently studying.
Our next question is, do these exosomes affect all cells the same way?
Yes, that was a very interesting thing that we had to think about, since we know that all cells secrete exosomes. Normal cells, healthy cells secrete them as well. So we've seen that the genetic makeup of the cell that receives the exosomes will play an important role in that response. So KRAS mutations are found in most pancreatic cancer cells, but there are some pancreatic cancers that don't have KRAS mutations. And we saw that those cells don't have a boost in chemoresistance from these fibroblast-derived exosomes. So it seems to suggest that not all cells will respond to this exosome in the same way, and we're still not clear how transformative the exosome from the calf can be. We're still early in looking at how normal cells might actually be transformed to full-blown cancer cells by these exosomes. But yes, so far our data suggests that they're very different response depending on the nature of the cells.
Thank you for that answer. Our next question is, what about epithelial cell exosomes? Do they play a role in this process?
Yes, we don't want to understate the importance of the epithelial exosomes. We know that they are there, especially in our in vivo experiment where we're combining the fibroblasts and the epithelial cells together. We know that the GW4869 is inhibiting release of exosomes from both the fibroblasts and the epithelial cells. What we saw in our in vitro analysis, and unfortunately I didn't have time to show this data, is that the cancer-associated fibroblasts, their protective effect seems to be higher even than the effect that exosomes from drug-resistant cancer cells actually have. And so we know that the cancer cell exosomes are important. That's why we use those exosomes as a control for all of our experiments, but it's just that the fibroblast-derived exosomes seem to be more potent in their effect. Whether that has to do with some other intrinsic value that these exosomes have compared to the epithelial exosomes is what we're interested in, but we do know that they have an effect, so we can't conclusively say, we wouldn't ever want to say that the exosomes from epithelial cells don't matter, because they do, but we just see a more potent effect from our fibroblast-derived exosomes.
Okay, we have time for one more question. Do proteins and exosomes matter for chemoresistance?
Yes, that's one of the things that we are investigating through another project in the lab. As I mentioned earlier, exosomes contain microRNAs, RNAs, DNA, and protein. We focus initially on microRNAs because microRNAs are what's enriched in exosomes. We do know that the proteins play a very important role. So what we did was a similar type of analysis where we performed mass spec on the exosomes from fibroblasts that were treated with chemotherapy or not, and looked at the profile of proteins that we found inside. And there definitely is some overlap in terms of the pathways, but it seems to be a very unique pathway also that seems to be more important for how the exosome proteins actually play a role in chemoresistance. And so it's, again, a fascinating field to look at how what's packaged in an exosome might matter. So we definitely are exploring in other studies the role of proteins as well, but we definitely believe they will play an important role as well.
I would like to once again thank Dr. Reginald Hill for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through January of 2017. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcements with your colleagues who may have missed today's live event. That's all for now. We thank you for joining us, and we hope to see you again soon. Goodbye.